Good morning, everyone. Welcome to the class of biology on the fascinating world of biology. Hi, I am Nashad. I will be your instructor for this course. Throughout this course, we will learn about digestion in humans. Now, let's start to learning skills. How? Here we will improve our reading skills. Now we discuss about digestion in humans. Look at this diagram and try to understand the process of digestion in human. Digestion in humans. What is digestion? Digestion involves two processes: physical digestion and chemical digestion. Physical digestion involves the mechanical breakup of food into small particles. This occurs in the mouth when you chew food by the action of the teeth and tongue, and in the stomach where continual contractions and relaxations of the muscles in the stomach wall cause a churning action. This breaks up the food particles and mixes them with digestive enzymes. Physical digestion increases the surface area to volume ratio of the ingested food so that digestive enzymes can act on the food more effectively. Chemical digestion is the breaking down of the large molecules in food, such as protein, starch, and fats, into small soluble molecules which can be absorbed. This involves a hydrolytic reaction catalyzed by digestive enzymes. Look at this diagram and try to understand the breaking down of large molecule into small molecule. Here is a large structures of protein plus water in the presence of hydrolytic enzymes, and these large molecules are break down into small molecules which are soluble like this. Small breaking down is takes place here. Soluble molecules, that's a chemical digestion. Processes that take place in the mouth. Food in the mouth stimulates the salivary glands to secrete saliva. Saliva is mixed with the food. Mucin in saliva helps to soften the food. Mucin enzymes here. Mucin enzymes soften the food. Saliva contains an enzyme called salivary amylase which digests starch to maltose. The pH of saliva is neutral around pH 7. Salivary amylase is active at this pH. Chewing breaks the food up into smaller pieces. This increases the surface area to volume ratio for salivary amylase to work on. The tongue rolls the food into small, slippery, round masses or bully, singular bullets. The bully are swallowed and passed down into the esophagus via the pharynx. Look here diagram and try to understand this or readings process. Is a mouth present and food is entered by mouth. Here are saliva present and upper side is salivary gland is present. In the mouth, chewing breaks up the food and mucin soften the foods. Salivary glands secrete mucin to soften the food. And bolus, the small piece food is entered by the mouth, and salivary glands secrete mucins which soften the food. The food is broken down by the teeth and salivary enzymes, and food, the small piece of single food is called bolus. Here is bolus shown. Food is rolled into boli and passes down the esophagus. Digestion begins in the mouth. Digestion starts in the mouth. The role of the esophagus. Peristalsis in the walls of the esophagus pushes each bolus of food down into the stomach. Gravity also helps the food to pass down the esophagus. Digestive processes that take place in the mouth. The presence of food in the stomach stimulates the gastric glands to secrete gastric juice into the stomach cavity. The presence of food in stomach stimulate the gastric glands. Gastric glands secrete the gastric juice into the stomach cavity. Peristalsis in stomach wall churns and break up the food. Peristalsis also mixes the food well with gastric juice. Gastric juice is a dilute solution of hydrochloric acid about pH 2 and two enzymes pepsin and renin. The dilute hydrochloric acid stops the action of salivary amylase by denaturing it, changes the inactive forms of the enzyme pepsinogen and prorenin in the gastric juice to the active forms. These are called pepsin and renin respectively. Provides a slightly acidic medium suitable for the action of gastric enzymes and kills certain potentially harmful microorganisms in food. The proteases, pepsins, and renin act on proteins. Pepsin digest proteins to polypeptides or peptones. Pepsinogen plus hydrochloric acid convert into pepsin proteins in the presence of pepsin polypeptides. Look at this diagram of stomach and microscopic views of the parts of the stomach. His small dots are branched in shapes cordillid proteins, and hairs are milk proteins, and the enzymes are renin, and these structures, the highly complex and complicated structure is protein, and hairs are the present of pepsin, and polypeptides bond is here present. These are the microscopic structure of the stomach. Proteins are partially digested by pepsin, and milk proteins are curdled by renin in the stomach. Here look at the diagram of stomachs and their microscopic views of the stomach. Renal clots are curdless milk protein by converted the soluble proteins caseinogens into insoluble casein. This is necessary because soluble milk protein would pass through the stomach to the duodenum as easily as water and would not be digested by pepsin. Insoluble casein remains long enough in the stomach to be digested by pepsin. Look here, prorenin plus as hydrochloric acid act as a catalyst convert into renin and soluble milk protein in the presence of renin insoluble milk proteins and insoluble milk proteins in the presence of pepsin convert into polypeptides. Food normally remains in the stomach for about 3 to 4 hours. The partly digested food becomes liquefied, forming chyme. The food which is chewing, grinding, and becoming to a mixture of liquid. This mixture of liquid is called chyme. Chyme passes in small amounts into the duodenum when the pyloric sphincter relaxes and opens. Here are the structures of stomach and food particles are present and the mixture, the smallest particle of food is called chyme. Chyme enter into the end part and fall into the duodenum. These are the part of small intestine which is called the duodenum. Chyme enter into the duodenum. Chyme pass in small amounts into the duodenum. I hope this lecture is very useful for those learners which are slightly weak in reading skills. Uh, uh, I hope this lecture is very useful for learner. Thank you for listening my lectures. We will meet in our next class, which is the topic of uh, digestion in the small intestine. Thank you so much.